Taste is one of our special senses, and it begins the moment you place food on your tongue. The chemicals in that food interact with clusters of sensory cells called taste buds, and these taste buds sit inside something called papillae spread across the tongue. Each taste bud contains several types of cells that detect sweet, sour, salty, bitter, and umami. Once they're activated, they send signals through 3 different cranial nerves before the brain interprets them, and the brain combines that information with smell and even the trigeminal system to form what we recognize as flavor. So how does the tongue actually detect taste? In this video, we are going to break the entire system down step by step. We are first going to talk about the macro anatomy of the tongue, take a visual look and understand the different papillae on the tongue, where they are found and which ones actually contain taste buds. Then we will zoom into the microanatomy of taste buds. We will go inside a taste bud and see the specialized cells that detect chemicals from food. And while doing that, we will understand the 5 taste modalities: sweet, sour, salty, bitter, and umami, and what each one actually responds to in detail. And we're going to see how taste is detected, the exact mechanism of how molecules dissolve in saliva, reach the receptors and activate taste cells, and of course understand the nerve supply and the pathway to the brain, which cranial nerves carry taste from different parts of the tongue, and how the signals reach the gustatory cortex. In the end, we will see how taste combines with smell and trigeminal input to create the full experience we recognize as flavor, and why food tastes different when you're congested. What's up everyone, my name is Time, I'm a medical doctor and I make animated medical lectures to make different topics in medicine visually easier to understand. If you would like a PDF version or a quiz of this presentation, you can find it on my website, along with organized video lectures to help with your studies. Alright, let's get started. Let's start by looking at the tongue from an upper view. Just for orientation, here we can see the epiglottis, lingual tonsil, the palatine tonsil between the arches, and we can see the terminal sulcus, which divides the tongue into an anterior 2/3 and a posterior 1/3. Now, when you look at the surface of the tongue, it's not smooth at all. It's covered in small bumps called papillae, and these papillae give the tongue its rough texture. Some of them help you grip and move food around, while others contain the taste buds that detect chemicals from what you eat. To understand how taste works, it's important to get familiar with these different papillae, because each type sits in specific regions and has its own role. There are 4 types. The vast majority of the bumps you see when you stick out your tongue are located across the anterior 2/3, and these are the Filiform Papillae. Near the tip and sides, you find the Fungiform Papillae scattered between them. Along the posterior-lateral border, there are the Foliate Papillae arranged as vertical folds. And right at the back, just in front of the terminal sulcus, you have a large Circumvallate Papillae lined up in a characteristic V shape. Let's now mark one section of the tongue and perform a little surgery by cutting this portion out, and then zoom in. We can now clearly see the different papillae and how densely they cover the surface. As you see, the vast majority are the Filiform Papillae. These are thin, cone-shaped projection that covers most of the anterior 2/3 of the tongue. Even though they are the most common, they don't contain any taste buds. They help you manipulate food, they increase friction, and they contribute to the rough feeling of the tongue. In many animals, especially in cats, these Filiform Papillae are much more pronounced and even keratinized, allowing them to groom, clean, and strip meat from bone. In humans, they are far smaller and purely supportive compared to those specialized functions. Alright, next, mixed in between the Filiform Papillae, especially near the tip and the sides of the tongue, are the Fungiform Papillae. These ones are slightly larger and rounder, and they really do resemble tiny mushrooms sitting on the surface. Each fungiform papillae contains a few taste buds on its upper surface. They are responsible for a lot of the taste sensations in the anterior part of the tongue. If you've ever noticed slightly larger reddish dots near the front of your tongue, that's essentially what you're looking at. Moving further back along the sides of the tongue, you will find the Foliate Papillae. These appear as a series of vertical folds. In humans, they are mostly active during childhood and gradually become less prominent with age, but they still contain taste buds located deep within the clefts between the folds. Even though they aren't as visually obvious as the other types, they contribute to taste detection along the posterior-lateral regions of the tongue. And finally, at the very back of the tongue, just in front of the terminal sulcus, there are the Circumvallate Papillae. These are the largest papillae and form a V-shaped line pointing towards the throat. Each one looks like a dome surrounded by a circular pit, and the walls of that pit are packed with hundreds of taste buds. Now, even though most taste buds are within papillae of the tongue, you can also find small numbers in the soft palate, the epiglottis, and parts of the pharynx and larynx. These don't sit inside papillae, they lie directly in the epithelium, and they primarily help detect bitter substances before swallowing. This is part of the body's protective system because it allows potentially harmful or unpleasant tasting chemicals to be detected even moments before they enter the esophagus and then down to your stomach. So when we put all of this together, the surface of the tongue is made up of Filiform Papillae, which provides texture but no taste, Fungiform Papillae near the front, which contains small numbers of taste buds, Foliate Papillae along the sides, which contains taste buds within their folds, and Circumvallate Papillae at the back, which contains large concentration of taste buds. And most importantly, all regions that contain taste buds are capable of detecting all 5 taste modalities. For many years, it was thought that the surface of the tongue has special areas for each of these sensations, but it is now known that all tastants are sensed from all parts of the tongue and adjacent structures. Alright, we've now seen the papillae from the outside, and we said that 3 of these papillae types, fungiform, foliate, and circumvallate, contain taste buds. Those taste buds are structurally the same, no matter where they sit, but their density varies depending on the region. That is why at the back of the tongue, especially around the circumvallate papillae, has a much higher concentration compared to the tip. Make sense so far? Good. Let's zoom into one of these papillae so we can finally see the actual organ of taste, the taste bud. A taste bud is essentially a small oval collection of 50 to 100 special epithelial cells responsible for detecting taste, called taste receptor cells or gustatory cells, and they are tightly packed together with a tiny opening at the surface called taste pore. This pore is simply where dissolved chemicals from food come into contact with the taste cell inside. And the dissolved part really matters because taste only works when molecules mix with saliva. If the tongue is dry, taste detection drops dramatically. Once something dissolves, it can diffuse through the taste pore and reach the upper parts of the taste cells. Inside each taste bud, there are 4 main cell types: type 1, type 2, and type 3. Plus a group of basal cells at the bottom. Type 1 cells are glial-like support cells. They help with structural organization and clean up neurotransmitters after signaling. Type 2 cells detect sweet, bitter, and umami taste using G protein-coupled receptors. Type 3 cells detect sour stimuli and form direct synapses with sensory neurons. At the base of the taste bud, we have the basal cells, which act as stem cells. These continuously divide and replace older taste cells that wear out, because taste cells actually live for only about 1 to 2 weeks. This rapid turnover is essential to maintain normal taste perception, because the receptors are constantly exposed to mechanical stress, heat, and chemical irritation from what we eat and drink. In addition to this, recent research as of 2023 has identified a specialized population of sodium taste cells that detect salty taste. We'll talk about this in detail in a moment. Now, we said earlier that there are 5 taste modalities: sour, salty, sweet, bitter, and umami. The sour taste is caused by acid, meaning by the hydrogen ion concentration, and the intensity of this taste sensation is approximately proportional to the hydrogen ion concentration. This means that the more acidic the food, the stronger the sour sensation becomes. The salty taste is formed by ionized salt, mainly by sodium ion concentration. Different salts taste slightly different from one another because they can trigger additional taste sensations beyond just saltiness. Let's look at table salt as an example. Table salt, or sodium chloride, has the chemical formula NaCl. When it dissolves, it separates into 2 types of ions: cations, which are positively charged, and anions, which are negatively charged. In table salt, sodium becomes the cation, Na+, while chloride becomes the anion, Cl-. The cation, especially sodium, are the primary source of salty taste. However, the anion also play a role in shaping the overall flavor, though their contribution is smaller. This explains why different salts produce distinct taste experiences. Sodium chloride tastes purely salt, while potassium chloride has a salty, bitter quality, and other salt combinations create their own unique flavor profiles. Sweet taste responds to sugars like glucose, fructose, sucrose, but so do a lot of completely different chemical groups. Things like glycols, alcohols, aldehydes, ketones, esters, some amino acids, and even certain small proteins. Many artificial sweeteners also fall into this category, despite being structurally unrelated to natural sugars. What's interesting is that even a tiny change in the chemical structure of one of these molecules can flip its taste response entirely, sometimes turning something from sweet to bitter. The bitter taste, like the sweet taste, is not caused by any single type of chemical agents. Here again, the substances that give the bitter taste are almost entirely organic substances. Two particular classes of substances are especially likely to cause bitter taste sensations. There's the long-chain organic substances that contain nitrogen and alkaloids. The alkaloids include many of the drugs used in medicine, such as quinine, caffeine, and nicotine. The bitter taste, when it occurs in high intensity, usually causes the person or animal to reject the food. This is undoubtedly an important function of the bitter taste sensation because many deadly toxins found in poisonous plants are alkaloids, and virtually all of these cause intensely bitter taste, usually followed by rejection of the food. Umami is a Japanese word meaning delicious, designating a pleasant taste sensation that is qualitatively different from sour, salty, sweet, or bitter. Umami responds to amino acids like glutamate, which is why foods such as tomatoes, soy sauce, cheese, and meat have a strong umami profile. So now the question is, how do these taste cells actually detect them? Sweet, bitter, and umami are all detected by G protein-coupled receptors located on the type II cells. When any sweet, bitter, or umami substances binds to its receptors on the type II cell, it activates a G protein called gustausin. This triggers the enzyme phospholipase C beta 2 to split a membrane lipid PIP2 into DAG and IP3. IP3 then opens calcium channels on the endoplasmic reticulum, releasing calcium into the cell. That calcium activates a channel called TRPM5, causing influx of sodium, and all that eventually leads the cell to depolarize. Type II cells don't use a typical neuronal synapse. Instead, they release ATP through special ion channels, and that ATP directly stimulates the sensory nerve fibers at the base. Sour taste is detected by type III taste cells, and the key trigger here is the hydrogen ion, H+, from acidic food. When acid enters the mouth, hydrogen ions activate OTOP1 channels on the taste cell membrane. These specialized proton channels allow hydrogen to flow directly into the cell. This proton entry does two things simultaneously. First, the positive charge carried by the hydrogen ions begins to depolarize the membrane. Second, the influx of protons acidify the cell's interior, and this drop in intracellular pH blocks the potassium channels called KIR 2.1. When these potassium channels close, potassium can no longer leave the cell, as it normally would, which further shifts the membrane potential and causes the cell to depolarize. Once depolarization reaches a threshold, voltage-gated sodium channels open, allowing a rapid influx of sodium ions that generate action potentials. These action potentials then trigger voltage-gated calcium channels to open. The resulting calcium influx is so important because the increase in intracellular calcium concentration triggers synaptic vesicles to fuse with the cell membrane through a process called exocytosis. This releases neurotransmitters, primarily serotonin and GABA, into the synapse where they stimulate the affected nerve fibers. That signal then travels to the brain and is perceived as sour. Now, salt taste is particularly interesting because the research on this has evolved dramatically. For years, scientists thought type I cells were responsible for detecting taste, but a major breakthrough in 2020, confirmed in comprehensive reviews published in 2023, completely changed our understanding. Salt taste is actually detected by a specialized population of cells that express both epithelial sodium channels called ENaC and calcium homeostasis modulator channels called CALHM13. These sodium taste cells are their own distinct sensory cell types. They don't really fit into one of the type 1, type 2, type 3 categories we just discussed. They're electrically excitable, which means that they can fire action potentials just like neurons. Now here's how it works. When sodium from food dissolves in saliva and enters through the taste pore, it passes through ENaC channels into these specialized salt detecting cells. That inward rush of sodium depolarizes the cell membrane. Once depolarization reaches a threshold, it triggers voltage-gated sodium channels to open, and these generate full action potentials. Just like in neurons, these action potentials then activate the CALHM13 channels, which release ATP directly onto the sensory nerve fibers below through what is called a channel synapse. This unique mechanism does not use traditional synaptic vesicles, and the entire process is incredibly fast because it runs purely on electrical signaling. Even though all these mechanisms differ, the basic arrangement is the same. The upper part of each taste cell samples the dissolved chemicals through the taste pore, and the lower part communicates with the sensory neurons. These neurons belong to the facial nerve, the glossopharyngeal nerve, and the vagus nerve, depending on the region of the tongue and pharynx. We will break down those territories in the next section. Taste buds also show variations depending on location. Circumvallate taste buds tend to respond strongly to bitter substances, which fits with their role as a final checkpoint before swallowing. Fungiform papillae near the tip often responds more strongly to sweet or salty substances, but even though these tendencies exist, each taste bud contains a mix of cell types capable of detecting multiple taste modalities. There isn't a sweet-only or bitter-only taste bud. Alright, now, food has entered the mouth. Saliva has broken it down into chemicals. The chemicals have entered the taste pore and have stimulated the respective taste cells, and that will now activate the nerve ending underneath the cell. Now we need to figure out how that information actually leaves the tongue and reaches the brain. Now, taste is not carried by one single nerve. The gustatory system uses 3 different cranial nerves, and each one is responsible for a specific region on the tongue and its surrounding structures. The anterior 2/3 of the tongue, this whole front portion, is supplied by the facial nerve. More specifically, it is carried through a branch called the chorda tympani. These nerve fibers enter the tongue along the lingual nerve from the mandibular division of the trigeminal nerve, but they separate once they reach the taste bud. The trigeminal part gives general sensations like touch and temperature, while the chorda tympani fibers carry only taste. So all the tastes coming from the tip of the tongue and most of the anterior surface travel through the chorda tympani and then joins the facial nerve. The posterior 1/3 of the tongue, all the taste information here is carried by the glossopharyngeal nerve, cranial nerve 9. This includes the Foliate Papillae on the posterior lateral sides and especially the Circumvallate Papillae at the back. Even though the Circumvallate Papillae lie technically in the anterior 2/3 by surface landmarks, they are innervated by the glossopharyngeal nerve. And because they house such a large number of taste buds, a major portion of our overall taste input actually arrives through the cranial nerve 9. The epiglottis, the upper part of the esophagus, and parts of the pharynx and larynx also contain scattered taste buds, mostly tuned towards bitter taste. And these areas send their taste fibers through the vagus nerve, cranial nerve 10. This provides a final protective checkpoint before swallowing. If something tastes intensively bitter or unpleasant at this stage, the body can still trigger gagging or reflexive avoidance. Alright, now I will test you. The anterior 2/3 of the tongue is innervated by cranial nerve number 7, called facial nerve. Specifically, which branch of the facial nerve? The chorda tympani. Good. The posterior 1/3 and the circumvallate papillae is innervated by the cranial nerve number 9, the glossopharyngeal nerve. The rest, the larynx, pharynx, epiglottis, are all innervated by cranial nerve number 10, vagus nerve. Awesome. Once these signals leave the tongue and upper airway, all 3 nerves converge in the same place deep in the brainstem. They enter the medulla and synapse in a nucleus called the nucleus of the solitary tract. This is the very first processing station for taste. From here, second-order neuron from the nucleus tractus solitarius projects upwards to a region of the thalamus, specifically the ventral posterior medial nucleus. This is the same thalamic relay that processes facial sensations, and taste has its own dedicated area within it. From the ventral posterior medial nucleus, third-order neurons travel to the primary gustatory cortex. This region is located in the insula and the frontal operculum, tucked deep within the lateral sulcus. This is where taste becomes a conscious perception. It is where you identify something as sweet, sour, salty, bitter, or umami, and where the brain begins to combine that information with smell, texture, temperature, and even emotional association. A portion of taste pathway also branches off at the brainstem level to control autonomic reflexes. For example, signals from sour or sweet food can stimulate salivatory nuclei, increasing salivation before you even swallow. Bitter or irritating tastes can activate gag reflex pathways. These reflex arcs run through the superior and inferior salivatory nuclei and are essential for digestion and protective responses. So, the gustatory pathway is essentially a three-step chain. First, taste buds, then cranial nerves 7, 9, and 10, then to the nucleus of the solitary tract, then thalamus, and then the gustatory cortex. Alright, so now we've seen how taste buds detect chemicals, how the nerves carry that information, and how the brain processes it. But here's the part that surprises most people. What we commonly call taste is mostly not taste at all. A huge portion of what you experience as flavor actually comes from smell. When you chew food, tiny molecules travel from your mouth up into the nasal cavity. These molecules stimulate the olfactory receptors, and the olfactory cortex then passes that information to the gustatory areas. So if you think something tastes sweet, fruity, smoky, or spicy, a lot of that comes from the olfactory input layered on top of the basic taste signals from the tongue. That is why when you're congested, food suddenly becomes dull and flat. The taste buds are still detecting sweet, salty, sour, bitter, and umami normally, but the smell component is missing. So the brain only perceives the most basic parts of the information. Remember during the COVID-19 pandemic that many people reported losing both taste and smell? In most cases of COVID-19, the taste buds themselves were not damaged at all. What actually happened was inside the nose. The virus affected the supporting cells around the olfactory receptors, the sustentacular cells, not the neurons directly. When those supporting cells became inflamed, the olfactory receptors temporarily stopped functioning. And because around 80 to 90% of flavor actually comes from smell, losing your olfactory input almost completely wipes out the flavor experience. The tongue can still detect sweet, salty, sour, bitter, and umami perfectly well, but without the smell component, the brain only perceives the simplest part of the signal. That is why everything tastes strangely muted, flat, or even unpleasant despite your taste buds working normally. There is also a trigeminal system, which is carried by the trigeminal nerve. This system does not detect taste or smell, but it senses things like heat from chili peppers, cooling from mint, tingling from carbonation, and the sharpness of wasabi or mustard. These sensations add another layer to what we think of as taste, even though they're technically separate from gustation. So when you put everything together, flavor is really a combination of three systems working at the same time. Taste from the tongue and epiglottis, smell from the olfactory epithelium, and sensations from the trigeminal nerve. The brain integrates all of this instantly, and that is why something as simple as a piece of chocolate can taste smooth, sweet, aromatic, and warm all at once. And with that, we've covered the next special sense in our list. I really hope this was helpful. In the next video, we will cover the next special sense, which is how smell works. I've made free courses on other topics here on YouTube if you want to keep learning. Otherwise, if you want a handmade PDF version of this lecture, take a quiz to test your knowledge, or access an organized list of all my videos, you can find everything on my website. Thanks for watching. See you in the next one.